So thank you for the introduction. And first I want to thank the organizers to give me the opportunity to speak about my work. So yeah, my title is Using Ancestral Sequence Reconstruction to Characterize an Allosteric Bi-Enzyme Complex. First, I want to show something about protein complexes. So 80% of all proteins form complexes, as it is here the case. And for complex formation, an interface is required. These interfaces are highly conserved due to the critical role. So this is shown here in a schematic way. So in the core region, the conservation is high, and on the rim region of the interface, the conservation is low. Within my talk, I'm concentrating on the protein complex imidazole glycerol-phosphate synthase, which consists of two subunits, this F and this H. Here the interface of this F is shown, and it's very similar to the schematic conservation of an interface. So here we have a high conservation in the core region, and to get more red with low conservation to the rim region. However, this is a well-characterized complex, but however, the relevance of interface residues for the complex stability is unclear. So we want to look on the binding behavior here now. So as we know, his F and his H from the same species are binding to each other. But form his F and his H from two different species also a complex. So we made our test case. We took his F from Pyrobaculum azenaticum, PA, and his H from Cymomonas mobilis, short ZM. So as we would expect, PA, his F and his H are forming a complex, and ZMSF and ZMSH also form a complex. But if we test the binding affinity between PASF and ZMSH, we found out that they have only a poor binding affinity, and so, yeah, they're not binding to each other. So we want now to address the question, which residues in his F are responsible for the different binding behavior? So we want to discover the binding hotspots. As we compare PASF and ZMSF, we have seen that they share only 64.4% sequence identity, and so testing all these mutations is unfeasible. So we got the idea to use ancestral sequence reconstruction, short ASR, to create a binding generalist and use this as a starting point. So we generated a phylogeny with phylobase, and then we reconstructed the ancestral sequences on the ancestral nodes with the program FastML, and here on this ancestral nodes we got the most likely sequence. Then we synthesized these ancestral proteins and characterized them experimentally. For the farmost ancestral node, we got the last universal common ancestor, short LUCA. So this LUCA's F showed in the wet lab that it is a good binder to ZMSH and also a good binder to PASH. So it is a binding generalist. So here now is the real phylogenetic tree for imidazole glycerol-phosphate synthase shown, and LUCA's F is now like an intermediate between ZM and PA. And if we now compare the two SF, so the LUCA's F and the PASF with this different binding behavior, we found out that they differ in 103 positions. Within the phylogenetic tree, we can now trace back the evolutionary path from PASF to LUCA's F to find also poor binding his F with more similarity to LUCA's F. And there we have found this ANK1 PASF, which now has a poor binding affinity to ZMSH and differ now only in 29 positions. So we minimized the candidate positions from 103 to 29. As the binding, as the interface residues are crucial for the binding, we looked on them bioinformatically and have seen So by comparing the interface of Lucas-F and ENC1-PAS-F, that these interfaces only differ in 9 positions. And then we assessed them biochemically and transferred these 9 interface positions from Lucas-F to ENC1-PAS-F, and now we got from the poor binding complex now a good binding affinities to ZMSH. And so we can say that these 9 positions are crucial for the binding to ZMSH. But what about this contribution of these 9 positions to the complex stability? So here we use the fold X analysis and wanted to assess the complex stability and the mutation. So we mutated not only single mutations, so the amino acid from Lucas-F and the other way around, but also multiple mutations, but also multiple mutations, so I think they will show it again now. And within this case, so so we expect that if we mutate in Lucas-F, we got a destabilizing effect, and if you mutate in ENC1-PAS-F, we got a stabilizing effect. And here now, there are the best scoring positions shown, which has the most stabilizing effect with mutated disposition in ENC1-PAS-F, and the most destabilizing effect when mutating these positions in Lucas-F. And here you see these combinations of mutations. Within these mutations, position 72 and 74 are overrepresented, so we looked on them biochemically again. So now we transferred only the amino acid from position 72 and 74 to ENC1-PAS-F, and we got a good binding affinity. So what about the single contribution of these 2 positions? First, we mutated only position 72, and we have still a poor binding affinity. But if we mutate position 74, we can get a good binding affinity to ZMSH. So this position is solely responsible for the binding affinity. If we mutate this position now in the already good binding complex Lucas-F-ZMSH, so taking the residue from ENC1-PAS-F to Lucas-F, we can disturb this binding, and we got a poor binding affinity. So we can say that position 74 is an interaction hotspot with respect to ZMSH. So now we have looked on this red evolutionary path, and to get the relevance of this hotspot, we also want to look on the blue evolutionary path. As we know here from the wet lab, is that TMSF and ZMSH are a weak binder. And the next evolutionary partner, ENC2-TMSF, is a good binder to ZMSH. If we mutate now here also the corresponding position 74 to the phenylalanine from Lucas-F, we can get a good binder. And if we mutate here in ENC2-TMSF, also this corresponding position 74 to an aspartate from TMSF, we can disturb this binding and get a poor binding affinity. And so this position 74 is also a hotspot for the interaction of TMSF. So overall, I can say that ancestral sequence reconstruction reduces the number of experimental binding studies, and in silico methods reliably predict the contribution of interface residues to complex stability. For precise determination, a biochemical characterization is required. Unexpectedly, this hotspot residue is not conserved and is located at the rim of the interface, so it has been not assessed so far with other methods. Combining the in silico and experimental approaches is an ideal combination to identify critical positions in protein-protein interactions. And within this, I want to thank my supervisor, Rainer Merkel, and Alexandra Holinsky for the biochemical contribution, and her supervisor, Reinhard Sterner, also the DFG, which funds my project, and all my group colleagues, and you for your attention.
questions from the audience? I have a question. So here you've talked about the analysis of binding sites or proteins binding to each other. So have you also looked at the active sites of enzymes? And, you know, can you say anything about critical residues or catalytic activity with your method?
So within this method, we compare one active and one not active one. So like non-binding and binding one. So normally these proteins were all active, so we couldn't do this comparative thing. But you could take it, so if you have a non-active one and active one, and if you compare it with this procedure, it would be able, I think, to identify a critical residue in the active site.
Okay, well then, thank you very much again.